Hello, everyone. Today, we're going to examine the third major virulence strategy, immune evasion. In previous lectures, we discussed how pathogens attach to hosts and how they invade tissues. But invasion alone is not enough. Once a pathogen enters the body, it immediately encounters host defenses. By the end of this lecture, you should be able to explain how the immune system recognizes microbes and describes several strategies pathogens use to avoid destruction. By the end of this module, you should be able to describe these common immune evasion mechanisms. And so, as a review from the host perspective, infection appears simple. We've talked about this. From the pathogen perspective, we've got this more complex process. First, we have to find an acceptable host to encounter. We must then adhere to avoid being flushed out. Then we need to usually establish a deeper invasion and colonization. Um, and now we're kind of getting to that point of how do we avoid detection now that we're inside the cell. So imagine a bacterium successfully attaching to a tissue and entering the body. At that moment, the battle is not over. It is just beginning. The host has multiple defense systems designed to recognize and destroy those invaders. Phagocytic cells engulf microbes. Complement proteins damage bacterial membranes. Antibodies bind foreign targets and help eliminate pathogens. For a pathogen to persist, it must somehow avoid all these defenses. Some hide, some disguise themselves, some directly interfere with immune functions. The examples we'll discuss today illustrate all three of these strategies. Brief review of some of our immune system that we've talked about already. Um, the innate immune system recognizes broad patterns that are common among microbes. These microbial patterns are called PAMPs, or pathogen-associated molecular patterns. Um, Also, you can have DAMPs, which detect damaged cells that were damaged by microbes. Examples of PAMPs include LPS, or lipopolysaccharides on gram-negative bacteria, peptidoglycan that's found in the cell wall, flagellin that's part of the bacterial flagella. Host cells contain receptors to detect these PAMPs called PRRs, or pattern recognition receptors. When PRRs recognize a PAMP, they activate inflammation and other immune responses. An antigen is slightly different. An antigen is a specific molecule recognized by the adaptive immune system, particularly B cells and T cells. So remember, PRRs recognize PAMPs. Adaptive immunity recognizes antigens. Both of those need to be avoided by the infecting microbe. One strategy is simply to hide. Uropathogenic E. coli, or UPEC, as we've talked about, can be taken up by bladder epithelial cells. Rather than being destroyed, some of the bacteria survive inside these cells. Once inside, they become much harder for immune cells and antibodies and complement proteins to reach. This intercellular lifestyle creates a protective niche where bacteria can persist even after symptoms improve. Hidden bacteria may remain and later reemerge, contributing to recurrent urinary tract infections. Now, this pathogen, Neisseria gonorrhea, has a different strategy. Instead of hiding, they continually change their appearance. The bacterium can alter proteins found on its pili and outer membrane. As the immune system produces antibodies against one version, new variants appear. The immune system is essentially chasing a moving target. The process is called antigenic variation. Because the surface structures keep changing, immunity after infection is often incomplete, allowing repeated infections throughout a person's lifetime. Another strategy is preventing immune cells from capturing the pathogen in the first place. Strep pyogenes produces an M protein that interferes with phagocytosis. It's the same M protein that's involved in attachment. The bacteria also produces a capsule that helps conceal surface structures from immune recognition. So if you remember the capsule, it's that polysaccharide layer that is outside of the cell membrane. Um, and Together, these virulence factors reduce uptake by macrophages and neutrophils. If phagocytes cannot effectively engulf the bacterium, the pathogen gains valuable time to multiply and spread. So, what have we learned here? A pathogen's job is not finished once it enters the body, and even after it's attached. In many ways, invasion is only the beginning. The immune system immediately begins searching for signs of infection and deploys multiple signs um, deploys multiple defenses designed to eliminate the invader. The innate immune system recognizes pathogens through pattern recognition receptors, which detect common microbial structures known as PAMPs, or pathogen associated microbial. Gosh, I just completely forgot what the last letter in PAMP is, which is molecular patterns. Oh gosh, forgive me, class. I lose my microbiologist card today. The innate immune system recognizes pathogens through pattern recognition receptors and detecting these PAMPs, which are these pathogen-associated molecular patterns. Silly. If a pathogen cannot overcome these defenses, the infection ends quickly. Successful pathogens have evolved strategies to avoid destruction. Some hide inside the cells, others make themselves hard to see, and then even others can just avoid being phagocytosed. And there's multiple examples of these, and there are different ways that different pathogens take on each of these strategies. Um, so, although these strategies are different, they all accomplish the same goal, increasing the pathogen's chances of survival inside the host. And as we continue studying pathogenesis, remember that disease is not simply a result of entering the body. The pathogen that cause disease are often the ones that can successfully avoid, manipulate, or overcome the immune response long enough to grow, spread, and transmit to the next host. With that, thank you for attending this lecture. See you in the next one.